So again, what I want to do with the second half today is to look at what nervous tissue is specifically. And this goes with some of the lab material for this section. And as I'm talking about the tissue, I want to show you some micrographs of the tissue, which is in next week's folder. I put the lab material in next week's folder. There's a link right here that says nervous tissue, which goes to a online folder with some captured images of the microscope slides. And so I'm going to be using those to talk about some of the tissue. There's a lab quiz here called the Neuroanatomy Lab Quiz. And it's in next week's folder. Its due date is set for before the following week. Again, I keep saying next week's folder, but of course next week we don't have class. That's 2 weeks from now. But what will probably happen is I'll spend all of next week dealing with the electrical activity stuff, and we won't get to the lab component of what I want to do for this week's material, the next week's material. So we might actually do that the following week. And if that happens, I'll change the due date for this lab quiz to reflect being due a week after we actually do the material. But that sort of depends on how I'm doing with covering the material I want. I'm actually going to be doing that material for the first time in my night class tonight. And then I'll do it again on Monday, well, for my Monday class next Wednesday, because they're doing that after the holiday. So I'll have done it twice already before we do it on, it's the 29th, right? They've already said. So maybe by that time I'll actually have gotten the presentation down to a more manageable way, and we'll actually cover that next week when we get to that week's material. But if we don't, then the Neuroanatomy Lab Quiz, which covers some of what we're going to talk about now, its due date will be pushed off until after we finish all of the neuroanatomy stuff.
Can you get extra credit if you do it early?
No. Because if you do it early, you're going to be trying to answer questions about information that you've not heard yet, and you might get it wrong. So it'd be the opposite of extra credit. It'd be a bad idea to try to take it early before learning the material. Okay. I've offered plenty of extra credit in this class at this point. Don't ask for more of extra credit. Okay. Anyway, so I want to talk about the tissue, and I'm going to include some of the micrographs that are in this link here. Now, to begin with, nervous tissue is composed of two different types of cells. There are neurons, which are the nerve cells, and I want to talk about those in a little bit detail before we move on to the other type, which are glial cells. And I'll talk about those when we get to that. Now, I've already put these pictures into other tabs so we can see them a little more larger. This is your standard kind of picture of what a neuron is in general. A neuron basically has a cell body and then things stuck to that cell body or sticking off of that cell body. The cell body is also called the soma, which is just the Greek word for body. And that's where we have the nucleus and the cytoplasm that contains all of the major organelles. Mitochondria, endoplasmic reticulum, Golgi apparatus, ribosomes, et cetera, all of that stuff's in the cell body itself. And then sticking off the cell body, there are a number of structures that collectively we refer to as processes. A process, anatomically speaking, is just something sticking off of something else. When we were looking at bones, certain bones had processes, as there was a spiky thing sticking off of a bone or whatever, that was a process. Here, when we're looking at a cell, anything that's sticking out from a cell is a process. they're not unique to neurons. Other types of cells have processes also. But when we're talking about neurons, the processes are either the axon or the dendrites. A neuron is going to have one axon coming off of it, and then everything else coming off of it is a dendrite. The dendrites are usually thought of as the receiving part of the cell. They get input from other cells. And then the axon is the sending side. So it sends information out to the next cell that it's contacting. In this picture, kind of like ghost images in the background, here's the end of another neuron making contacts with the dendrites here, and then the axon that we're looking at, it ends up making contacts with dendrites of another cell. And those contacts are called synapses. We talked about one type of synapse already, the neuromuscular junction, but there are more types of synapses, or more examples that we'll talk about next time. Also, what we see in this picture, wrapped around the axon are these kind of, I don't know, clearish blue-green looking things, which are called myelin sheaths. Myelin is an insulating substance. The myelin is actually a component of another cell, which is drawn in here. And so this cell wraps its cell membrane around the axon and provides insulation that way. Kind of like if you're making pigs in a blanket, you take dough and wrap it around a hot dog or Vienna sausage, whatever. Same idea, just wrapped around this quite a bit. And I'll show you some pictures later on when we're talking about glia that explains that a little bit better. These little insulating packets, these myelin sheaths, are not a continuous insulation. There are gaps in between them every so often called nodes of Ranvier. That's their full name. Ranvier is the last name of the scientist that they're named after, and he was French, so Ranvier is how you pronounce it. I'll often just refer to them as nodes when we're talking about them, just for the sake of saying it quicker, but the full name is node Ranvier. And those gaps, those nodes, are important in how the electrical signal is sent along the axon, which again we'll deal with next time in more detail, but we see them here. So that's a basic structure of a neuron. These three cartoons are showing us some of the variety of neurons that we have. Now, the first way that we classify neurons is based on what we call their polarity, or how many things there are sticking off of the cell. So we have a unipolar neuron, and the cell body only has one thing coming off of it, which is its axon. And then a bipolar neuron has two things coming off of it. One thing's its axon, and the other thing is a dendrite. And then we have a multipolar neuron, which is more than two things coming off of it. One is an axon, and all of the rest are dendrites. Now, as this cartoon's drawn, it's kind of similar to this sort of picture we're looking at here. But actually, multipolar neurons have a lot of different kinds of shapes to them. Unipolar cells all kind of look the same. Not exactly like this cartoon, but along these lines. There's this one big cell body, and then it has a thing sticking off of it, that one axon sticking off. The axon splits into going two directions. Unipolar neurons are pretty much always found carrying sensory information from your periphery into your central nervous system. So one side of the axon ends with dendrites, and those would be the nerve endings in the skin, for instance. And when those nerve endings are activated by sensory stimuli that interact with the skin, an electrical signal is sent along the axon past the cell body, and at the end of the axon, it starts branching out to reach out to all its different targets. like we see here. This is, again, kind of a cartoon representation, but they're all sort of laid out the same way. Bipolar neurons also don't have a lot of variation in their shape because really it's a cell body in the middle with two things sticking off of it, and they're usually going in opposite directions, 180 degrees apart. One side is the dendrite and one side is the axon. So one side's receiving information from one place. That information goes down the dendrite across the cell body and out the axon to eventually reach its target, wherever that might be. And then multipolar neurons come in a lot of different shapes and sizes. They're called multipolar because there can be any number of things sticking off of them. Unipolar has one, bipolar has two, multipolar has three or more. So we could have one axon and two dendrites. That'd be a multipolar neuron. Or we could have this that has one axon and all of these dendrites sticking off of it. How many dendrites are sticking off of it doesn't really matter, just as long as it totals with the axon more than two things sticking off of it. And we're going to look at some examples of multipolar neurons in tissue to give us a better idea of what they look like. And this picture is the next one in the chapter. And I want to use this for a second just to talk about the variety of shapes. And then we'll look at some of these in the tissue. The first two are examples of multipolar cells. So we can see the cell body in the middle there, the cell body in the middle there, and there are definitely multiple things sticking off of it. And there's, as it's drawn here, only three things sticking off of this, but it's more than two, so that counts. The first one is what's called a pyramidal cell, and it's named pyramidal because the cell body is kind of triangular in shape. So a triangle in 2 dimensions. In 3 dimensions would be a pyramid or a cone, depending on its exact shape. But somebody called it a pyramidal cell because they thought it was pyramid shaped. It has one dendrite sticking out of the top and then a bunch more dendrites sticking out of the base of the pyramid, which we see there. And then the axon comes out right at the middle of the base of the pyramid. The other multipolar neuron that we see in the next part is called a Purkinje cell. It's named after the scientist who discovered it, which he was from Czechoslovakia, which isn't even a real country anymore. It's essentially where the Czech Republic is these days. So Purkinje is a horrible mispronunciation of the Czechoslovakian name that I think is pronounced something like Purkinje or something. But everybody calls it a Purkinje cell. It has this one axon coming off the bottom that we see here, and we see a couple of branches off of that axon because it has several targets. This target is going to a neighboring cell. This target is somewhere far away. We just see the axon end. We don't see it's sort of cut off. We don't see where it finally ends. And this branch seems to be going up into this area right here. It kind of looks like it synapses back on itself, which is possible, but I can't think of any examples of that for Purkinje cells. Other cells will do that. But so this is another example. So those are the different things that axon can do. Just one axon leaves a body, but it goes to different places. And then we see 2 major dendrites coming off of this cell, and they go on to branch to make a lot more different branches, filling up sort of all of the space above the cell in the picture. Okay, so these 2 are very different looking cells, but they're both multipolar cells, and they're both multipolar cells like this, which looks also very different. Okay. Over here we have bipolar cells down here. These are olfactory cells, meaning that they're responsible for the sense of smell. The dendrites are sticking out at the bottom of the picture, which is actually sticking out into the nasal cavity from the nasal epithelium. And so odor molecules will interact with the dendrites here, causing signals to be sent up through the axon where it synapses actually in the brain. We don't see the bone drawn in here, but there's a bone separating the nasal cavity from the cranial cavity, and the axons go through that bone to synapse, and then cavity there. There's really only two examples of bipolar cells in the nervous system that we're going to be dealing with at all. One of them is these in the olfactory system. The other one is found in the retina of the eye. Both of those are special senses, and so that's really a topic for A&P2. So other than pointing them out in this little picture, we're not going to deal with bipolar cells anymore this semester. But also in this picture, the synapses from the bipolar cells onto these dendrites right here follow back to a cell body right here that has a couple of more dendrites sticking off of it and an axon extending away from this, and we just see it cut off, but it goes farther into the brain. So these have four things sticking off of them, which makes them multipolar. But again, they look different from the other multipolar cells we have here or this multipolar cell that we started by looking at. So multipolar cells have a lot of different shapes, and all that we need to say to consider something multipolar is that it has more than two things sticking off of it. And that's what we see in all of these examples. Okay. Now, what I want to do to talk about neurons a little bit more is actually switch to the micrographs and talk about it a little bit first. Now, the first type of cell I want to show you is actually this type of cell, kind of close to what this drawing represents. They're found in the spinal cord. And so here is a micrograph of part of a spinal cord. And let me turn the lights off so we can see it there. This is not the entire spinal cord. It's a cross-section of the spinal cord, but it's only part of it. Actually, let me pull up a picture real quick. So what I'm showing you is a part of a section like this. Just to orient you to what we're seeing here, the spinal cord is this kind of oval structure here. It has white matter, which is the lighter staining part here, and gray matter, which is the darker staining part. There's a hole right in the center, which we see here, and then kind of a line extending along the actual midline of the spinal cord, and then we kind of have a mirror image of the structures on one side versus the other. What we see coming out of the spinal cord up here, down here, and coming together, this is part of the spinal nerve. So this is actually peripheral material. This in the middle is the central nervous system stuff. The picture I'm showing you, which I'm going to go back to in a second, is basically a field of view that just shows this much of this particular section. So kind of imagine a rectangle inset right here, and that's what we're looking at. So here's that hole in the middle with a line going up and down, showing the midline of the whole thing. And then the slightly darker staining gray matter versus the lighter staining white matter here. So the whole thing is much bigger than what we have on the picture here, but we're looking at a segment of it. What we're interested in looking at here, the particular neurons that I want to point out, are these big dark blobs that we see down in the gray matter here. These cells are essentially shaped like the picture that we have here. Not precisely, but that's what this drawing is based on, what they look like. And so we want to look at them under a higher power to get more detail. So I have a spinal cord gray matter slide at 10x and 40x, so we can see a little bit more detail. When we go up to the 10x objective magnification, we start to make out the individual cells. And the thing about how we're staining these neurons, they don't look exactly like what we would expect. So the nucleus is actually lighter than the rest of the cell. And that's just a quirk about how this staining works. And there's a little dot in the middle of the nucleus, okay, which we see, for instance, in this one down here, these two up here, this one over here. We can see the void of the nucleus in this one or this one, a hint of it there, a hint of it there, but no dot in the middle of it, just because the dot is a little spot in the middle of the nucleus, which picks up a lot of stain. And if we don't cut through the exact right place in the cell, we're not going to see that little dot. Now, what that little dot is a nucleolus. The word means little nucleus in Latin. What it actually is it's the region in the nucleus that codes for ribosomal RNA. And ribosomal RNA is what ribosomes are made out of. So also in that area, it's where ribosomes are assembled. So back to our picture here. That little dot that we see there, that's picking up a lot of stain because it's where the ribosomes are found and where they're being assembled. Let me move from the 10x objective to the 40x objective. And I've zoomed in on that same same cell I was just pointing at. And we can see the little spot where the nucleolus is within the greater nucleus. This staining technique actually stains RNA best. So we don't see the DNA in the nucleus very well, but we can see where the RNA for ribosomes is being made in the nucleus. And then what we're seeing in the cytoplasm out here are all the ribosomes after they've been assembled. They've moved out into the cytoplasm of the cell. So this is a cell body. The staining of the cell body looks kind of patchy because the ribosomes are going to be clustered in particular areas. When you think of ribosomes in a cell, what do you usually associate those with?
Protein synthesis.
So functionally, yes, they're important for protein synthesis. And where do they mostly do that? Yeah, the rough ER. So these little clusters are actually showing us where there's a little region of rough ER. Now, when we're talking about protein synthesis associated with rough ER, what that usually means is those proteins are being directed towards particular parts of the cell. It could be that they're going to be secreted out of the cell, which is not terribly common with neurons. More often with neurons, they are proteins that are going to be incorporated into the cell membrane. So say a cell that's making insulin will package up insulin in a vesicle and release its contents outside the cell, and it'll secrete the insulin. For these, it does the same sort of thing, but instead of containing stuff within the empty space of a vesicle, the proteins are in the cell or within the membrane wall of the vesicle. And when the vesicle joins with the cell membrane, like it was secreting its contents, instead its contents will be part of that membrane and therefore get incorporated into the cell membrane. Okay. The kind of things that neurons make like this are going to be ion channels and neurotransmitter receptors and other proteins that have to do with chemical and electrical signaling between neurons. So there's a lot of ribosome activity in these cells, which we can see by staining. We see a lot of ribosomes because they're making lots of proteins that have to be sent and incorporated into very particular parts of the cell. Now, these cells that we're looking at, we've actually already heard of. These are the cells that make skeletal muscle fibers contract. So last time when we were talking about motor units, I defined a motor unit as one neuron and all of the muscle fibers it connects to or controls. This is one of those neurons. So the cell body is found in the spinal cord, which would be, let's say, at this level of spinal cord, in my lower back region, the lumbar region. Its axon is going to leave the spinal cord and extend all the way down to its target axon. And so I'm picking out one down here because these neurons control muscles in the lower extremities. This cell, if that's what I'm talking about right now, might contact a muscle in my big toe. Okay, so its axon is going to go all the way from the lower back down the length of my leg to the ankle, turn around the ankle, and go all the way out the length of my foot. For me, that might be 1 meter in length. Okay, depends on how tall somebody is, exactly how far that distance is. But so this big cell body is necessary to make all the proteins necessary for the entire cell. We're seeing other neurons here that are different sizes. Okay. Now this little thing off to the side here, this little thing off to the side here, look like they're smaller neurons, but probably what it is it's just a neuron of a similar size that we're cutting sort of the edge off of, so it sort of looks smaller. Imagine if you're slicing up a tomato. Okay, as you're slicing the tomato from the edge, you get smaller pieces of tomato, and they get larger and larger as you get to the middle of the tomato, and then they get smaller and smaller again as you get to the edge, if you slice tomatoes. So this is sort of like a slice at the edge of the cell, which isn't the entire size of the cell. This is closer to the center of that cell. So that's kind of what the size of that cell. The smaller cells we see actually are these things like this. Okay? This has a pretty small nucleus compared to the nucleus of the cell I've been pointing at. And this might be part of its cytoplasm. Okay? So this is not a very big cell compared to this one here. And it probably only contacts cells in its local neighborhood. So it's a small cell because its targets are all nearby. The big cells are the ones that have targets that are far away from the cell body. Okay? And actually, we're going to see that sort of as a theme. We're going to see these big cells. And for the big cells, they're big because they're associated with connections that are very far away. So there's long axons or there's big dendrites or something like that associated with it. And the cell body is where all of those proteins are being made for all of those parts of the cell. So the bigger the cell body, the more proteins that are being made, that sort of thing. Now, I was talking about, whoops, wrong picture, the shapes of cells earlier. You're not going to need to worry about what shape a particular cell is because the bipolar cells, like I said, that's really from senses that we're going to talk about in AP2. So we won't deal with those at all this semester. Unipolar cells are only found in one place as far as we're concerned in this class. And that's going to be in the next slide I show you. Everything else we look at is a multipolar neuron. Okay? So if we're looking at this picture, this cell here, we can see there's definitely something sticking off here and something sticking off here. There's probably something sticking off there and maybe even something sticking off there. So we can see 3 or 4 things sticking off of this cell. Okay? Or we can see where 3 or 4 things are sticking off of it. So this is definitely multipolar. And even the one I'm showing you here, there's surely something sticking off there, something sticking off there, and maybe there. So 3 or 4 things sticking off of that too. It's definitely multipolar. But if we go up here to this sort of thing, maybe there's something sticking off of this side, maybe there's something that's sticking off of this side, which makes it look kind of like it might be bipolar. But it's not. All of these are multipolar cells. It's just a matter of where you actually look at or where you cut through the cell. Okay? If we're cutting through this cell and we're seeing all of these things, we're getting through kind of the center of the cell. But if we're cutting through this cell and we only see a couple of things that might be sticking off of it, we're probably cutting through the edge of this cell. Actually, I want a different picture. So here's a number of different micrographs showing us these kinds of cells. This one is probably a good example of what I'm talking about. The slide that we're looking at, where'd that go? Here, we're looking at a very two-dimensional image. Okay, it's a very thin section and very shallow field of view, focus. So we're not getting any three-dimensional shape to this. But this picture I'm pulling up here, this is a thicker section. We're getting more stuff in the field of focus, or the plane of focus. So we can see, for instance, a couple of dendrites sticking off and even crossing over each other. So they're probably going in kind of different directions. And it looks like there's something in the back here, and this might be something kind of towards the back, and this is sticking out towards the back. So this has a little bit more three-dimensional shape. Okay, if we cut a section directly through this picture, following where I'm drawing this line here with the finger, we wouldn't see any of those things sticking off of it. Okay, we might not be able to say it's multipolar if we're cutting right here. If we cut, say, from here up this way, we might see 2 or 3 of the things sticking off of it. But we really need to appreciate its three-dimensional shape to see its multipolar character. And we're just not going to see that in these kind of sections. So you're not going to be looking at these and trying to decide whether they're multipolar or something else. All of the cells we're looking at under the microscope are multipolar, except for the ones I'm going to show you in the next slide. We won't be looking at any bipolar cells under the microscope, or in the microscope slides that I have here. So if I ever ask you what shape a cell is, it's likely to be multipolar. And the exception to that is when I'm showing you or talking about this next type of cell, which is the unipolar cell. So I was just showing you the 4x objective picture of the spinal cord, and then the 10x and 40x objective of the gray matter of the spinal cord. I'll show you the 40x objective picture of the white matter when we get to talking about glia, because that's where glia are. Now, to show you the unipolar cells, I'm going to show you this thing called the dorsal root ganglion at 4 and 10x, and then a 40x objective that's labeled unipolar cell down here. This is a ganglion. This is outside of the spinal cord, but it's actually very close to the spinal cord. It's attached to the nerve root that goes into the spinal cord. Now, I pulled up this picture earlier because this is a good picture to see what I'm talking about. So here's our spinal cord cross-section that we were just looking at. Coming off right here, this is a nerve root, and this is also a nerve root down at the bottom. The way that this picture is oriented, the bottom of the picture is anterior, the top of the picture is posterior. Anterior and posterior have alternate names for them. Posterior is also called dorsal. So the nerve root that's attached to the posterior or dorsal side of the spinal cord is this one right here that I'm pointing at. If we follow that nerve root back, we see this little bulge on the nerve. This is the dorsal root ganglion. Now, at this magnification, we can't see any detail in it. But the picture I'm showing you here is a higher magnification of that kind of structure. We can actually see one cell really standing out here. We see a really dark spot in the middle, and then the lighter staining cytoplasm of the cell around it. I'm going to zoom in on this to the 10x objective, and I'm pretty sure I'm zooming into this area right here. I can't remember precisely where I picked, but I think that cell I was just pointing out that's really dark is this one right here. Okay. So we can see that cell again here, but now we can see more cells. There's a big cell there, smaller cell there, another cell there. There's a few up here. There's a nice big one there. A few that go off in a stream here. It's a little cluster down here. A ribbon of them going across here and going up here. They're sort of all in these little clusters here. The space between these cells and these cells, all of this in between, looks like a bunch of sort of wavy lines. And those are the axons of the nerve that this ganglion's attached to. And in fact, those axons are axons of these cells here. These are unipolar cells. And there's one thing coming off of each of these cells that goes to give rise to the fiber that's passing through, the axon that goes through this. We can't see that one thing. The chance of actually seeing it in a section is pretty slim because it's only one thing sticking out of the spherical shape of the cell. So we don't really see anything sticking out of them, but that's what's there. Do you have a question, Emily?
When you say, okay, this is the ganglia, like, what do you look for that identifies that to you? The fibers?
Ganglia are where there are cells.
Okay.
So there are cells in this structure. The cells have fibers associated with them. So these fibers are the axons from this, but the fibers continue on in this direction and continue on in this direction. Okay. Going back to this picture, this is the ganglion. Okay. And so some of those fibers are continuing on here as the dorsal nerve root. And then for this picture, it's cut off, but more are going to be continuing on out in the other direction. And that's the nerve that's out in the periphery. Okay. It's going to connect to the skin or muscles or wherever it happens to be going out there in the periphery. That's what we're seeing there. So these fibers are fibers that make up the greater nerve that extends around it. The cell bodies that are clustered at this point, that's the ganglion around it. Okay. So if we go to the, whoops, wrong direction. Oh, that's right. The 40X objective is down here. I've named this specifically unipolar cell instead of dorsal root ganglion, but it's just a higher power of the dorsal root ganglion picture we're looking at. So here's one of those cells, and I can't remember at all which of the cells out of the slide I was just showing you it is, but there's its nucleus and then the rest of its cytoplasm. These are not staining exactly like the ones that I showed you in the spinal cord. The staining is being done a little bit differently, but we can see the structures of the cell here, nucleus, cell body. That's a really big cell body. Okay. These other things we see around it here are cell bodies of other types of cells, and there are nuclei we see of cells out here in the bundle of fibers around it. So this is a really big cell, and again, the big cells usually indicate that it's supporting a lot of other structures, and for these unipolar cells, what they're doing is they're carrying sensory information from some structure in the periphery, like our skin, and sending that into the central nervous system. So again, kind of like I was talking about with the motor unit neuron that I was showing you a second ago, that might control a muscle in my big toe, this ganglion might be located at the base of the spinal cord here, the lower back region. Its axon might be coming from the skin in my big toe. The axon goes up past the cell body of the ganglion, and it continues on into the spinal cord, and it actually doesn't synapse with anything until it gets up to the brainstem right up here. So from the tip of my big toe to the brainstem up here, that's one axon that's supported by a big cell like this. Okay, so while the motor neuron that I was showing you earlier, I said its axon might be about 1 meter long for me. This is probably 2 meters long because it's going sort of the whole way between the big toe and the brain. With the motor neuron, it's just going from the spinal cord out to the muscle, so it's only half as long. Okay. But they get to be really big cells. Not all of the cells in here are necessarily that big. So here's a couple of smaller cells next to some of the bigger cells. And how big they are just corresponds to how long their axons are. So some of the ones that are connecting to structures that are closer by might be smaller cells, or they might be carrying different information that doesn't go all the way up to the brain. It might go just into the spinal cord. So there's some variation in size, again, corresponding to where their connections are. So this slide, these dorsal root ganglion slides, which the 4 and 10x objective modifications are here, and the 40x objective is labeled unipolar cell down here. That's the unipolar cell. That's the only micrographs we have of that. Everything else we're looking at, neuron-wise, are multipolar neurons. And the only thing we've looked at so far are the, where'd it go? Spinal cord cells, which we're just looking at. So we can see the little thumbnail that came up there. And that's what we were just looking at. I want to show you a different type of multipolar neuron, and this one's found in the cerebellum. Now, the cerebellum is part of the brain. The word cerebellum actually means little brain in Latin. Let me take this little half of a brain model out of the dorsal here. When we think of the brain, we usually think of this big wrinkled outer surface here. This is what's called the cerebellum. This is what's called the cerebrum, which just comes from the Latin word for head or brain. This little thing here is a cerebellum. Okay? And it's actually attached back here in the brain. So it's this little version of the big brain that's in the back, called the cerebellum. Both the cerebellum and the cerebrum have wrinkled surfaces to them. So in this micrograph, what we're looking at is a section of the cerebellum, and we see cuts through the wrinkled surface. So here's the outer surface of the brain, and it goes in and it wrinkles here, and the wrinkle even goes folds in a little deeper, and then the wrinkle comes out here, and we have another sort of fold of tissue over here. Within one of these sort of wrinkled little pieces of the tissue, there's a fairly light stained area from the edge of the tissue into here. Now, all of this is gray matter, okay? So where we are in the brain, the gray matter is on the outside surface. This portion of the gray matter doesn't stain very darkly because it doesn't have a lot of cell bodies in it, but it is still part of the gray matter. But then the darker part that we see down here, which is still gray matter, is staining dark because it has a lot of little cells in it. Those little cells look like grains of sand. So this is called the granular layer of the cerebellum with all these grainy cells in it. In the center of the granular layer, you can see this little lighter streak. This is actually the beginning of the white matter that's deep inside the cerebellum. But what we're interested in is really where the lighter staining and the darker staining layers come together. Okay, right at that edge, there are a few really big cells that really stand out. So this is using the 4x objective. If we go to, oops, wrong direction. Oh, where is it? I thought they were next to each other. So I was just showing you the 4x objective of the cerebellum. So here's the 10x objective of it. So again, right where the darker staining, grainy looking granular cells come up against the lighter staining part here. There are these really big cells right on the border. These are the Purkinje cells that I showed you earlier in this picture here. We're just looking at the cell body part right there. That's what's at the edge. But what takes up all of that lightly staining part are all of the dendrites of that cell. So here's a couple of cell bodies. Here's another one. The dendrites extend out to fill up all this light staining area here. And in this slide I'm showing you, we can see the edge of the light staining part right there. So from where I'm pointing here all the way down, this whole region is filled with dendrites of these Purkinje cells. Okay. Their axons go down into this area here through the granular layer. A branch of the one I was showing you before would synapse on one of these granular cells, but then it would continue on to where that pale little region that I call the white matter is, and the axon would continue on for a while from there. But these are the cells we're talking about. Here's a 4DX objective picture of two of those cells. The nucleus and the nucleolus is visible in both of the cells, not quite as visible in this one. And then the cytoplasm is picking up some stain. In this one, there's a little bit of a hint of a dendrite coming off here, but it doesn't go very far. And here we can maybe imagine a dendrite starting to come off there, or maybe starting to come off there. Okay. But the dendrites don't stain in these slides because we're not using a stain that marks dendrites. It's just marking the cell body. But we can look at other slides showing different types of stains for Purkinje cells. Now, first, this picture here actually comes from a rather classical anatomy study that was done probably a little over 100 years ago now. There's this rather famous Spanish anatomist who loved to draw pictures of things, and I'm pretty sure that's what this picture's from. And he was able to stain these cells using something else. He actually used a chemical stain that was based on photographic chemistry. And so this is actually stained with a silver metal stain. And it fills up the entire cell, and we can see it very clearly. And he went in here and using a microscope, drew a picture of everything that he saw there. Okay, and we can see the axon coming off the bottom here. And then these are probably two different perspectives of the same kind of cell. They have a really broad array of dendrites sticking out, but that tree is fairly narrow if you rotate it a little bit. But that tree takes up all of that space that's the lighter staining parts. This micrograph here is stained kind of like ours, but a little bit more detail. So you can see the bigger dendrites sticking out. And so these cells all lined up on the edge next to the granular cells sort of fill up all that area. But we're not seeing as extensive branching pattern in here as was drawn here. Or there are other examples like this, where the cell has been stained with a fluorescent dye. Now there's actually this picture here, I kind of like what happened. This thing is a small glass needle that's injecting the dye into the cell here. And what that dye does is it gets into the cell and basically fills the whole thing up. And we can see every little bit. So this is sort of the modern equivalent to the photographic version of staining that the Spanish guy was doing hundreds of years ago, or 100 years ago. But they're just, prettier with this nice green color here. So there's a few variations on this. This is a computer reconstruction of that kind of staining, as is this with some different colors. I think the color coding here is getting at the diameter of the dendrites. So red for the thick parts and moving towards blue for the really thin parts there. There's another example. This one here shows that same kind of fluorescent staining, but they've stained a whole bunch of cells, so you can see how their dendrites kind of all overlap and completely fill up that area that's the lighter staining part of the slide I was showing you a second ago. So all of this light staining part in here, right now we don't see anything because the stain doesn't show us dendrites. But from what we're just seeing in those other stains, we can imagine that all of this area is filled with dendrites of this particular cell here. And again, it's a big cell because it's supporting a lot of structure. And in the case of the Purkinje cells, it's that it has all of those dendrites. Okay? All of those dendrites need lots of proteins, one, to give them their structure. Also, there needs to be ion channels and neurotransmitter receptors and all those sorts of things attached to the membrane. So big cell making all of the protons for the rest of the neuron. The axon coming off of it is also relatively long. Now, what I was talking about before with the things associated with the spinal cord or the ganglion, those are really long, 2 meter long axons. This axon is probably measured in centimeters, a couple of centimeters long or something like that. But compared to the width of the cell, which is, what would that be? Probably about 100 or 200 micrometers in diameter. 2 or 3 centimeters is a very, very long distance. Okay, I have no idea if this comparison is entirely accurate, but imagine that you were to walk as far as that dendrite extends away from that. So if you were a couple of 100 micrometers in width and you had to walk 2 or 3 centimeters worth of distance, that might be, and I'm actually kind of completely making this up, but to give you a sense of scale, that might be like walking from here to Holyoke or Northampton or Hartford or something like that. It's quite a distance given the scale that we're working at. Okay. If you were to walk as far as the dendrite, as far as the axon of a motor neuron in the spinal cord, that might be walking from here to Atlanta or something like that, if my scale's at all realistic. Someday I'll have to do the math to actually see if that's true. But so the distance that axons go might be pretty short in our scale of things, but compared to the size of the cell, it is rather a long distance. In comparison, the granular cells that are in here, these little dots, these are neurons too, and they're very small because they have just the barest little fringe of dendrites around them, and their axons might go from, we're talking about a cell here, its axon's only going to go up here, okay, which is measured in millimeters or a fraction of a millimeter. So that's not very far for this little cell. So it's a small cell, just supporting its a little bit of axon or dendrite in its local neighborhood. But big cells have lots of things taking off of them, or their axons go a long distance. So those are the Purkinje cells of the cerebellum. I would also kind of like to show you an example of pyramidal cells. We don't have a micrograph or a microscope slide that's very good of that. So for that purpose, I just want to go here and pull up some pictures. Sort of like what we were just looking at. This is a kind of nice picture. This has been stained with that kind of silver stain I was talking about earlier. And it's a great way to do this type of neuron. So you can see it's got a sort of triangular body, cell body shape. And some of the dendrites are coming off the base of that pyramid. And then one dendrite extends straight up with branches extending out in either direction. Here's a couple, well, this looks like a drawing, but these are probably drawings made from cells that are stained that way. That's a drawing too. This is what those types of cells would look like in the kind of stain that we are looking at, okay? So we really just see the cell body and maybe a little bit of the top dendrite sticking out. But we don't see the really broad dendrites, and we don't see the axon coming out of it. Now, I'm not using a micrograph from our slide set to show you, because we don't have a slide that's even this good. And I'm not crazy about this because it leaves a lot of stuff out. The slides that we have are stained more like this, and even not as well as this, but it's kind of hard to really make out all of what's in there. Nope, that's a cerebellum. Cerebellum. Here's another. These are more pyramidal cells here. Here's a pretty low power one of it. So they just look like these little flecks. If you get in closer, you can see the triangular shape of the cell body, but not a whole lot more than that. So we don't have really good slides in our set to show you, and these Google search versions are better to kind of point out what they're like. And just as I was scanning down here, I noticed here's the picture from the book here. I'll have to look into that some later on, but yeah, so that picture here from our book. Okay, so now I want to go on to talk about the glial cells. Glial cells, or sometimes they're just called glia, are called that because the name comes from the Greek word that means glue. Originally, they were just thought to be the cells that stick all of the neurons together. Neurons were appreciated early on as the important cells of the nervous system. But there are actually a lot more glial cells, maybe 10 times as many glial cells as there are neurons in the brain. And since there are so many of those cells, they must have some important functions to play. They just don't necessarily perform the primary function that we're interested in, which is the signaling part. We classify the glial cells based on whether they're found in the central or the peripheral nervous systems. And I've made two lists here, and I've lined them up so that similar cells correspond in this table. In the central nervous system, we have these cells called astrocytes, and in the peripheral nervous system, we have cells that are called satellite cells. Now, the name of the two is kind of related. Astrocytes are star-shaped cells, and satellite cells are described because they look kind of like moons or satellites orbiting a planet. So they kind of both come from, an astronomy point of view, what they're named after. They both are responsible for supporting the nervous tissue. Now, there's a lot that goes into support. I've just simplified it with a very basic term there. But the kind of things that they do are to regulate the concentration of ions in the extracellular fluid, to help regulate the use and reuse of neurotransmitters. And then the astrocytes especially are involved in what's called the blood-brain barrier, which is abbreviated BBB, because the brain and the spinal cord have a barrier separating them from the blood. Floating around in your blood right now could very well be some toxic molecules. We ingest all types of toxins with the food that we eat. There are toxic molecules in plants, and we eat, vegetables and that sort of thing, so there might be some things there. They aren't that toxic unless you get them in really high concentrations. But because they have the potential to be toxic, we don't want them to get into the brain. Now, that's kind of on an everyday basis, you're dealing with that sort of thing. But the blood-brain barrier is also going to help protect against real threats that might get into the body, whether it's pathogens or other kinds of foreign invading things. They can't get into the brain themselves. We also don't want things to get into the brain that are just normal chemicals in our body, because they can have very different effects in the brain. So the blood-brain barrier only lets small molecules like water and ions and those kinds of things through, as well as nutrient molecules that are necessary, which would be carbohydrates like glucose, amino acids, and lipids. Other than those things, nothing can get across the blood-brain barrier. And part of what keeps that from happening is the astrocyte. The astrocyte is usually associated with blood vessels, physically touching the walls of blood vessels and helping to regulate what can cross over and get into tissue. There's not really a blood-brain barrier kind of thing in the periphery, although satellite cells do try to protect the peripheral neurons from some of the things that are in the blood. So there's a very weak sort of barrier in the periphery within a ganglion. The next pair, oligodendrocytes and Schwann cells, are found in the central and peripheral nervous systems respectively, and they're both responsible for insulation, which means that these are the cells that wrap around the axons to insulate the electrical activity that's traveling along. That insulation is called myelin, which I mentioned earlier. So myelination is just another way of saying insulation. The picture of the neuron that I was showing you earlier here, the cell that's providing that insulation to this is an oligodendrocyte. Okay? Oligodendrocyte, we can translate very literally to mean few branched cell, okay? Or a cell with a few branches. So we have the cell body here, and the way this is drawn, there's a couple of things coming off of it. There could be a couple more or so, but very few branches coming off of it. And those branches extend out and wrap around part of the axon here. Okay? In this drawing, there are 5 little segments of myelin sheath. 2 of them are directly part of this oligodendrocyte. The other 3, we don't have any indication of where they're attached, but other oligodendrocytes would be providing those bits of insulation. Okay. And we'll talk about the structure of myelin in a little bit detail in a sec. Okay. Schwann cells in the periphery do a similar kind of thing, but the entire cell wraps around the axon. It's not a branch off of a cell that wraps around part of an axon. And I'll show you a picture of what that looks like in a little while. Now, the next 2 rows in this table, there's only an entry under the central nervous system. There's no entry in the peripheral nervous system because there's 2 other types of glia that are only found in the central nervous system and don't have counterparts in the peripheral nervous system, because in the peripheral nervous system, other tissues serve the same functions. These special types of glia are in the central nervous system because of the blood-brain barrier. The first type, called microglia, are responsible for immune surveillance and phagocytosis. That function in the periphery is handled by the immune system, by white blood cells. White blood cells can't get across the blood-brain barrier, so microglia act like white blood cells in the brain and spinal cord. Okay? And then the other type of cell, called an ependymal cell, is mostly associated with creating what's called cerebrospinal fluid, CSF. Because blood doesn't get into the brain directly, like it can interact with the tissues elsewhere in your body, these ependymal cells, some of them, are responsible for filtering stuff out of the blood and creating this cerebrospinal fluid, which acts as the fluid that's sort of integrated in with the brain tissue and the spinal cord tissue. They actually do a few other things, and I'll show you some pictures to depict what they do. What they do is kind of most closely related to epithelial tissue in other parts of the body. So since elsewhere in the body we have epithelial tissue that do epithelial things, the periphery doesn't need a special kind of glia to do that. But in the brain and spinal cord, we need ependymal cells to serve that function, which has a little bit to do with the blood-brain barrier also. Now this picture here shows a little bit of a rendition of what's going on there. Here we go. So here's an astrocyte, here's an oligodendrocyte, here's another oligodendrocyte, a couple more astrocytes, some microglia, these fuzzy looking things here, and then ependymal cells there. Now the astrocytes interact with neurons directly. There's not a blood vessel in this picture, but some of these astrocyte processes might interact with blood vessels. There's a suggestion of interaction with a microglial cell there. The oligodendrocytes have a few branches, and the two that are depicted here have 4 each, going out to wrap around part of these axons. There are other myelin sheaths represented here, but their particular oligodendrocytes aren't depicted here. This oligodendrocyte, we can see, contributes myelin to this axon in 2 places and to another axon in 2 places. And this one kind of does the same thing as it's drawn here. The ependymal cells are all lined up in a row because what they're doing is they're lining a little bit of space inside the brain, okay, or spinal cord. So they kind of look like an epithelium in that they're a single row of cells, but they interact with the other cells in the tissue here in a way that epithelial cells don't. Because again, they're not really epithelial cells. If I go to the, which one? Oh, this 10x objective of the spinal cord, this is highlighting that hole in the center of the spinal cord that I pointed out before. And the dark staining row of cells that basically lines that hole down the middle of the spinal cord, those are ependymal cells. Okay, so that's what they do. They line that cavity there. Now, this is in the spinal cord. There are similar structures in the brain that are lined with ependymal cells. And in some places in the brain, those ependymal cells are making cerebrospinal fluid. These cells don't make cerebrospinal fluid because it's not made in this spinal cord. It's made in the brain. And we'll talk about that in the next chapter. So that's going to be a few weeks off before we get to that. But those are what ependymal cells look like. In the white matter or the gray matter here, some of the smaller cells, so this is a big neuron here, some of the smaller cells in here are glial cells. We can't tell what are astrocytes and which are oligodendrocytes or microglia, although microglia are probably not going to be present in a small region like this. We can guess that the oligodendrocytes are going to mostly be in the white matter and the astrocytes are mostly going to be in the gray matter. But that's not 100% true. Okay? There might be an oligodendrocyte or two in the gray matter, and there might be astrocytes in the white matter. But I have another picture, that I'll show you in a little bit, that's spinal cord white matter at 40x objective magnification. And I'll go to that after we look at some other things. And what I want to talk about before we get there has to do with, this is the periphery kind of picture. So satellite cells are covering the outside of peripheral ganglion neuron, which would be a unipolar cell. And then Schwann cells wrap around the axon, providing insulation to that. Okay. So I want to show you what myelin looks like. We're actually going to look at a Schwann cell, which would be out in the periphery, but basically what happens with the Schwann cell is what's happening for oligodendrocytes also. So here we have an axon, and the glial cell, in this case the Schwann cell, starts to wrap around that axon. And it starts to tuck one end of its cell membrane around the axon and wraps around it a bunch of times. And those layers and layers of cell membrane that we see there that are labeled myelin, that's the insulation. Okay. There are proteins in the cell membrane there that contribute to the function of myelin, but collectively the cell membrane and the proteins in there is referred to as the myelin. In the picture down below, which let me enlarge this a little bit, this is an electron micrograph showing us a bit more detail of the layers and layers of cell membrane wrapping around this. Now, this is an electron micrograph, which is different from a light micrograph, just that the microscope uses electrons instead of light particles or photons to see stuff. Electrons are very small, and the only way to see something based on them is to block them from hitting the receptor screen where we see the image. And what blocks electrons are large atoms, and large atoms are metal atoms. And so there's two stains that we see here. You can't tell them apart color-wise because there's no color associated with electrons. But one stain that we use is osmium. Osmium is a heavy metal and it blocks electrons. It associates with lipids. So all of these layers here, these are layers of cell membrane. We can see them because osmium is attaching to them and blocking the electrons from hitting the screen. And then the other stain that we use is lead, and lead associates with proteins. And so a lot of these little things that we see inside the axon or in the tissue outside, connective tissue, or this is a nerve, so this is some periphery. There's some connective tissue there, and there's some other structures. A lot of what we see there, the little dots and stuff, are proteins. This structure that we see right here, and another one off to the side here, these are mitochondria. Mitochondria have a lot of membrane in them, so they stain with the osmium, but they have a very distinctive appearance under electron micrograph, so we can see that very easily. But what we're really interested in are the layers and layers of cell membrane there, okay? Now this magnification here, I think the book says it's like 1.5 million times magnification. So this is really, really high power using electron micrograph. I have some light micrograph images that I want to show you that are stained with osmium also. Now this is just stained with osmium. There's no lead here. So we're just staining lipids. The nerve is what we see up in the top corner that kind of looks like a slice of watermelon. All of this other stuff here is other tissue that's outside of the nerve. Now I'm calling this a nerve, which means that we're in the periphery. And what type of tissue in the periphery do you think would stain this way when we stain it with a heavy metal that marks lipids? What kind of tissue do you think we're looking at here? Yeah, adipose tissue. So when you looked at adipose tissue before, when we were studying tissues, it looked very light and kind of like spider webs. Now we're sort of seeing a reverse stain of it, and it's really these dark globules where all the lipids are. Lipids don't stain with the typical type of cell stain we use, so it was very light. But here we see a lot of dark staining where the lipids are. Okay. And this adipose tissue is just adjacent to the nerve, helping to kind of cushion the nerve as it's passing through wherever this might be. If we look at a higher power image of just the watermelon kind of shape here, we can start to see cross sections of axons that are wrapped in myelin, like these things down here, that really look a lot like that electron micrograph I was just showing you here. Okay. So imagine this, instead of at 1.5 million times magnification, at 400 times magnification or whatever this might be. So it's a lot less detail, but we can still see the same sort of shape. And it's that osmium that's binding to the lipids. So we can see the myelin wrapped around the axon. And the clear space in the middle, that's where the axon is, which isn't picking up any stain here. Okay. So this gives you a little bit of an idea of what the myelin looks like when we stain for that. We have the more traditional cell stain version in this spinal cord white matter 40X. Now we're staining for the cellular components. So for instance here, the dark thing in the middle, that's the axon, what was blank in the middle in the previous picture. And then what's blank here surrounding the axon, this is the myelin. Now this stain that we're using doesn't stain lipids, it stains the hydrophilic components of the cell. So we see the axon, we don't see the myelin. This is just a negative image of what we saw a second ago here. Okay. So if you imagine this sort of in reverse, what's dark here is what's light in that white matter, just because we're using a different type of stain. Another picture I want to show you is this thing called a teased nerve here. Before I go to that though, the watermelon shape that I keep describing, I use watermelon because a lot of students have called it the watermelon slide in the past. It's a nerve that's been cut in half, okay, sort of like a banana split. And then that half has been sectioned and cross-sectioned. So we're looking at a cross-section of half of the nerve. This picture, we're actually looking at a nerve that's been prepared in a similar way, and that the nerve has been cut open. But instead of sectioning it, we've teased out individual fibers. It's still stained with the osmium. So the black that we see is osmium staining the lipids of the myelin. Okay. Now, when we look at these axons, every so often we can see a little gap in the myelin like that. That's one of those nodes of Ranvier. Okay. And because we're staining for the lipid and not staining for protein or anything else here, it really stands out. There's a gap in between two sections of myelin. Okay. There's another one right here. There's another one right here. That might be one there. That might be one down there, et cetera, et cetera. There are a few around here. That's one right there. This is not a node of Ranvier. This is where the axon's been crushed a little bit. And so the myelin has been damaged, and it's not, it's either not there to be stained or it's not reacting with the stain appropriately. So that little gap there is not a node of Ranvier. This is a node. And then if we follow the myelin along, the next node that we see is right here. If we keep going, we don't run into another node. If we keep going the other direction, we don't see another node in this fiber here. Or in this fiber, we see a node there. If we keep going this direction, we don't see another node. If we keep going this direction, we don't see another node. This fiber is covered up by the other fiber crossing over here, so I don't know if there's a node there. But definitely in that much distance, we don't see a node, or in this much distance, we don't see a node. Okay. The point I want to make here is that the nodes are actually quite far apart from each other. When we look at a picture like this in a textbook, or you might see it online somewhere, the nodes are very close together, and the myelin sheaths look very short. It's an unfortunate necessary aspect of drawing a picture like this. To fit everything in, we got to play with a scale a little bit. So we make the distance from one node to the next look very short compared to the diameter of the axon or the diameter of the whole cell. But actually, based on the diameter of an axon, the distance from one node to the next is huge. So if we go back to this picture, if there was really the correct length of myelin sheath, this first one would extend off of this page. We'd never see the next node in the drawing. So for the sake of the drawing, they play with the scale a little bit, and they look really close together. Just keep in mind, in real life, that's not how close together they are. It's just for the sake of a drawing that we do it that way. But we can see it here. So I showed you the spinal cord and the dorsal root ganglion, the cerebellum, the ependymal cells. Oh, that's one more thing I need to show you. Back here to the unipolar cell, which is the ganglion at high power. The cells that we see surrounding the unipolar cell, these are the satellite cells. Okay? And they're called that because they look like they're orbiting around this bigger cell in the middle. And then the cells that we see in the fibers here are probably Schwann cells. Now, this is in the periphery, so it's satellite cells and Schwann cells instead of astrocytes and oligodendrocytes. But the Schwann cells are wrapped around the axons themselves. So their nuclei are going to be right in there with the fibers, which we see there. Okay. We can't be positive that all of the nuclei in the fiber region here are Schwann cells, but most of them probably are. Okay. So looking across all of these different micrographs, we can see examples of a few different types of multipolar cells. The one example, which is kind of the only example of unipolar cells. And then we saw the various types of glia represented as best as they can be in different locations. So going through all of these slides, we can see all of the different types of cells that make up nervous tissue. So that finishes up the nervous tissue part of the chapter. What we're going to go into next is looking at the function of this nervous tissue, concentrating on how electrical signals are sent from one cell to the next. So the action potential is that electrical signal, and then communication between neurons is getting at the synapses, where one cell passes its signal on to the next cell, kind of like we saw in the neuromuscular junction. So that's going to be the main point of what we look at next week. And like I said, I'm not positive by the time, again, it's not next week, 2 weeks from now. By the time I get to doing that for you guys, I might have this down to half of the class, and then we'll get to look at the rest of the neuroanatomy lab stuff. If I don't get to doing that with you guys, the neuroanatomy lab quiz due date will be pushed back a week because we'll be having to deal with the material a week later anyways. So we'll see how that plays out when we get to the next class. And again, our next class is 2 weeks from today on the 29th, okay? 14 days from today. And we'll finish that up, finish this chapter up that week. And then the next week on Wednesday and Thursday, we'll be finishing up all the rest of the stuff, looking at the anatomy and function of the nervous system and some of the clinical aspects of things in terms of the neurological examination that's performed in the clinic. Okay, so that's where we're going to go from there. That's all that I have